As usual, I'll do some announcements and reminders before we officially start, since it'll be on video for anybody who misses it. Good morning, Newton. Excuse me. So I finally caught up with my online classes with the grading and emails. So now I just need to get caught up with you guys. I apologize for the delay. But it will happen, and I'll let you know once it's done. So speaking of which, like for attendance, I still haven't even addressed all there's still emails that you guys have sent for attendance that you haven't heard back from me yet, and that's why. It's not that it's lost or that I'm ignoring you. I just haven't gotten there yet because I do it in order of which I've received it. So I'll let you know when I'm caught up. Anyway, what are the other announcements? Lab will go on as usual today. For those of you who are in the Monday's lab, as you know, the pH thing didn't work. I guess if you wanted to show up today to do the pH thing, you could, but again, you don't have to. And hopefully I'll do an online version of that. Again, for those of you in the Monday section who couldn't do the pH experiment, I'll give you the option of doing a virtual online version. It'll be sort of extra credit, sort of. Anyway, if you have any questions about that, ask me later. Let me go ahead and present now. It's 8 o'clock. We are starting chapter 3. We're done with chapter 2. So I'm gonna do what I've usually been doing, which is, I'll send you the links to the study guide, the links to some videos that are reinforcing what we learned in chapter 2, and maybe some stuff that we're about to learn in chapter 3. All the usual. Current slide. Anyway, like I said, it is 8. I'm gonna wait till 8:01 before we officially get started. Are there any questions while we're waiting for 8:01? There we go. Hopefully everybody can see just fine. All right. Whoop. So yeah, what's some other announcements? Don't forget to read when I post announcements. Don't forget to read them. It's been a while since I've done it because I've done so many announcements recently, and I apologize for that. But as you guys, as I've mentioned, sometimes in those announcements, it'll say something like, if you read this, reply to the email and tell me, and you get extra independent work points. So a lot of you either don't care about getting independent work points or you're just not reading the announcements. I don't know which. But yes, please read those announcements. And again, I try to keep them few and far between, but it is what it is. And if you guys have suggestions, if you have a priority or if you would prefer like a lot of smaller announcements versus fewer bigger announcements, let me know. I'm here for you. Anyway, it is 8:01. We'll get started. The first term, it won't even be a word. First term for today for attendance will be 2-hour delay because there should have been a 2-hour delay today. So again, if you're online sometime before 9 a.m., send me these attendance keywords. 2-hour delay is the first one. If you're watching a video, I don't know. In this case, just send me the word because there's no real screenshot for that. So just send me the word 2-hour delay or the term. Anyway, chapter 3, molecules of life. So we finished chapter 2. You learned all about chemistry. Well, you scratched the surface about chemistry. So you know about molecules and covalent bonds and ionic bonds and hydrogen bonds, and you know the basics. And hopefully, if you understand that, then you'll be able to understand chapter 3 a little bit more, which is where we're going to look at specific types of molecules. So not just molecules in general, like all that stuff you drew in lab on Monday or that you're going to draw in lab today. We're going to specify some molecules that are specific to biology, and they're important to biology. And to truly understand this, again, you kind of truly need to understand. Please let me know because I need to make sure you understand that so you understand this. You need to understand this chapter to understand most of the next, let's see, chapter 5, chapter 6, chapter 7, chapter 8, chapter 10. So at least the next 5 chapters. Basically, most of the semester, you're not going to truly understand it unless you truly understand this. So let's jump into it. Let's talk about the biological molecules. Anybody guess what this picture? All right. So of all the different types of biological molecules you may have heard of, what kind of molecule? What do you think they're trying to get at with this picture? I guess focus in on this, but the fact that they're including the human might also help. What do you think of when you think of meat? When it comes to like different types of biological molecules. Of course, I haven't introduced those terms yet, but I just want to see if anybody can guess. Good, protein, and that's exactly what your book was getting at. Yes, protein, and also your hair is made of protein, right? So I think your book is kind of getting at that. Like, there's an example of protein. So you're going to learn about what makes up protein and different things about protein in this chapter. And again, it's going to be really important because proteins pretty much can control how our cells work. So all the stuff we're going to learn later, to truly understand, you need to understand proteins. Does anybody know what this kid's eating? No, it's poop. No, I'm just kidding. Yes, it is chocolate, right? And I think your book was just getting at with this picture. You know, I don't know if you guys can tell, but the kids, you can see the meltiness, right? So the chocolate's melting. And your book is just talking about how certain things can be solid at room temperature, but then they become more liquidy at once they get a little bit warmed up. Things like fats, saturated versus unsaturated fat. And you're going to learn all about that in this chapter. And here, if I remember correctly, and I don't know if I am, someone can correct me if I'm wrong, I think they're just talking about DNA. So if you were to look at, the molecules of DNA, like your DNA, and compare it to an elephant or a bacterium or a protist or this grass, right? All the DNA on Earth is the same. Now, granted, the codes are going to be a little bit different. Like where I might have an AACG, he might have an AACT, and it'll be a lot different for the elephant. But the point is, the molecule itself is the same, it's just put together different ways. So anyway, let's move forward. I'll remind you, every chapter we do starts with this, biology and society, and it kind of introduces you to the theme of the chapter. The theme of this chapter is lactose. As always, I'm going to skip it. There might be questions about it on the study guide, so I highly recommend reading it and coming to me if you have any questions about it, but I'm not going to ask any questions about it on the exam. So let's just move forward. You will learn what lactose is a little bit later on in the chapter. We're going to talk a little bit more about it. If you have questions, again, about what lactose intolerance is, let me know, but that's not anything that I'm going to test you on. So here we go. This chapter is pretty simple in a way because there's only two main bullet points. We're going to talk about organic compounds and large biological molecules. So basically, the organic compounds discussion is going to be a generic discussion. So everything we're going to talk about when we talk about these different large biological molecules, they're all organic compounds. So before we talk about the different types of large biological molecules that you're going to need to know the differences between, we're going to talk about them in general, right? So here we go. Organic compounds. Something you definitely need to know from the last chapter, and here it is again, is that your cells are mostly water, right? Most of your cells are mostly water. And if you remember from last chapter, I said I'm not big into numbers, but I gave you a number that I said you should know. I should have put it on the screen again here, but it's 70 to 95%. Of course, that number varies so much because it depends on, well, first of all, it depends on the species, right? So obviously we're humans, so I'm discussing humans, but it would be different depending on the species. And then for you, it would depend on what kind of cell we're talking about. Brain cell, liver cell, all the different cells. And then how hydrated are you? All those different things. But yes, cells are mostly made of water. Now for this chapter, that's not an important fact. What your book is getting at here, the reminder here, is that once you take away the water, right, most of the cell contains carbon-based molecules. And this is just an introduction. I'm not necessarily going to ask you this. Other than water, what are your cells mostly made of, right? So I'm not going to necessarily ask you that. Hence the X. This is just an introduction as to why you're going to learn about these organic compounds and what they are, right? And your book points out that carbon forms large, complex, and diverse molecules. So for those of you who have already done Lab 1, as I'm sure you're familiar with from having to draw those molecules, carbon bonds 4 times, right? So if anything, really, that's what you need to take from that lab. All the hoops you have to jump through. The most important thing that you need to understand, the carbon bonds 4 times, nitrogen bonds 3 times, oxygen bonds twice, hydrogen bonds once. Not that I'll ever ask you that in a test question, but to truly understand the stuff I'm going to teach you, that does help. So yeah, like I said, carbon bonds 4 times. As far as we're concerned, there is no molecule that bonds more. I mean, no atom, no element, but we don't talk about it, right? So of all the elements that we talk about, all the bonds that we talk about, carbon's Carbon's the big one with its 4 bonds. Because again, it has 4 bonds that can do all kinds of stuff that the other ones can't do. Or as your book puts it, again, it can make large, complex, diverse molecules that are necessary for life. So again, if you've done the lab or you still need to do it, when you're drawing those molecules, this is why I always recommend starting off with the C's, right? Draw all the carbons and then put everything around that. 1, because they're organic compounds, so they're based on the carbon. And 2, because they have those 4 bonds, you can move things around them a lot easier. Anyway, so that brings us to the definition of what an organic compound is. It's a carbon-based molecule. I might ask you the definition of that, but probably not. Because like I mentioned a little bit earlier, there's 4 main types of molecules I'm going to be teaching you about, and that's what you need to know. You need to know the 4 different types, how they're similar, how they're different. So I'm probably not going to ask you the definition of organic compounds, but there you go. Now you know. So it has nothing to do with the organic section at your grocery store, right? It just means carbon-based molecules. Anyway, any questions about this slide? Relatively unimportant as far as the exam is concerned. It's just an introduction to what I'm about to teach you. Oh yeah, so for you guys who came in late, make sure you send me an email to let me know that you came in. Otherwise, I'll forget that you came in late, and I'll just have you as absent. Anyway, let's talk about some carbon chemistry, which I've already introduced you to in the last chapter, and I've really introduced it to you in the lab. As I already said, carbon's a versatile molecule. Why is it versatile? It's because it makes 4 covalent bonds, which is more than we talked about, like I just mentioned earlier. And because of that, it can make an endless diversity of carbon skeletons. As a matter of fact, skeletons will be the next word for attendance. Obviously, if you're in person and want to get the extra credit, like I'll just reinforce this, just send me the words sometime before 9. If you're online live and need attendance points, send me the words sometime before 9. If you're watching the video, I'm changing it up on you. Instead of taking a screenshot of the word skeletons, send me a picture of the word skeletons. Look it up, Google it. So yeah, for those of you in person and online right now live, everything's going as normal. But again, if you're watching the video, send me a picture of skeletons. Don't even send me the word. Just send me a picture of skeletons. And yes, I'm leaving that vague. Human skeletons, dog skeletons, cat skeletons, cartoon skeletons, whatever. Just send me, let it be yours. I mean, not your skeleton, your choice of skeletons. Anyway, these endless diversity of carbon skeletons, they vary in size, they vary in branching patterns. Don't need to write that down. Again, I'm still just giving you an introduction to what this whole thing is. The next slide I'm going to show you shows you some book examples of the varying size and branching patterns. So again, I'm going to move forward because other than the fact that carbon makes 4 bonds, this slide's not important for the exam. This is just kind of introducing you to what you do need to know. Don't write any of this down either. These are just examples. But here's a very basic carbon skeleton, right? 2 carbons and a bunch of hydrogens. That's it. Very simple, very small. Then you've got this one. It's a lot like that, except it's a little bit longer. And if you wanted to, just imagine one being even longer than that, because some of them do. Some of them are very, very long. Of course, not only is it a little bit longer, but you can see here that carbon, those 2 carbons have a double bond between them. So they have fewer hydrogens around them. And that'll be important later. Not that exact example, but that idea. Then here we have this one. This one forms a ring, right? That's very different. This one has some carbon, some double bonds, some single bonds. This phallic looking thing right here, we have, not only is it a chain of carbons this way, but there's also one going this way. And again, we have the hydrogens around it. So I'll ask again, how many of you are chemists? None, right? Just double check it, which is perfect. So probably you don't look at this, you can't look at this and understand what the chemical properties of these molecules are, right? You're probably not looking at this and thinking, I know what this happens. I know what happens when this thing mixes with water. Or I know what happens when this thing mixes with oxygen, right? You don't know those things, and that's okay. But this goes back to what I taught you in chapter 1. One of the themes of biology throughout the semester is going to be form and function, right? So you can tell by looking at these things, even without being a chemist, that they have different shapes, right? And a lot of times, chemistry, especially in biology, it really is a lot of times how it just fits together, how things fit together. So you can just tell by looking at this that these things are going to fit together differently, right? So they must do completely different things, and you would be correct. And as far as I'm concerned, that's all you need to know at this level of biology. That because we have these different shapes, and then we'll talk about the chemistry behind it later, because we have all these different shapes, these carbon molecules do such different things. And that's what your book's really getting at for that side. So any questions about that? All right, moving forward. As you already know, carbon can bond with a lot of things. Most commonly, and I'm going to present this like I did in the last chapter because this is how I'll ask about it on the exam, the big four, right? So you have carbon, hydrogen, oxygen, and nitrogen. That's mostly what we're going to talk about. Obviously, there are exceptions, and we'll even have some of those exceptions. Usually bonds with hydrogen, oxygen, and nitrogen. Again, when you're working on these things in the lab, I recommend doing the carbon, right? So let's say you had something that was C3. We'll just go ahead and do the C. And maybe even, let me change it up a little. So then also do the 4, so we know the carbon can bond. So when you're drawing your molecules, put your carbon, however many there are, and then fill in the blank. Because, if you haven't had lab yet, you'll see me do this in lab. If you've already had lab and you still need help with it, meet me during office hours. Anyway, any questions about the big four? All right, moving forward. Here's another example. No need to write any of this down. This is just an example. This is like the most basic organic molecule. It's called methane. It's CH4. Obviously, that means it's 1 carbon bonded to 4 hydrogen atoms. Your book points out that it's one of the most simplest organic compounds and that it's abundant in natural gas. And how's it produced? Well, by prokaryotes in swamps. So that's what we call swamp gas. And when I say prokaryotes, I'm talking about things like bacteria. And of course, in digestive tracts for grazing animals. So that's all stuff to look up if you want to for independent work, if you're interested in that. So methane is also a greenhouse gas. Your book doesn't talk about that. So yeah, the meat industry is actually adding to our greenhouse gas situation. Anyway, look it up if you want. None of this is going to be on the exam, just an introduction. So are there any questions about methane? All right, here's some different ways of drawing it. There's a structural formula. So again, for lab, this is what you need to be familiar with. These are the kind of things you're going to draw. But again, like I said, in the last chapter, I showed you some other versions of molecules. There are different ways of drawing things. So again, when you're back home and you're talking to your favorite chemist friend, and he's like, I know how to draw methane now, and you draw that, and he says, he draws this, and then you get to arguing. Don't argue. There's different ways to draw molecules. And again, as just an example, I'm not going to show you a picture of methane on the exam and say, what is this molecule? Or I won't say, draw me a ball and stick model of methane. So just examples. So any questions? All right. This is also just some examples. This will be a little bit more important when we get into chapter 5, but right now this is still an introduction to the stuff you're going to learn. So right now is not necessarily anything to write down, but your book does point out that it's those larger organic compounds that are the main fuels, the main molecules in gasoline. So when you think of gasoline, does anybody know what it's called? Well, it's right there in front of me. Never mind. Octane, right? So it's a long organic molecule. It's a bunch of carbons with hydrogens. That's it. And if you look here, this is dietary fat. And if you were not a chemist or not a biologist and you had to describe how is dietary fat like octane, I would say, well, dietary fat kind of looks like 3 octane molecules attached together. Of course, it's not exactly correct, but sort of. And sometimes that's good enough because, like I said, there's a relationship between form and function. So the fact that this thing looks a lot like this, it means something because they do the same thing in a sense. You're breaking down that molecule to release energy to do work. Of course, in this case, it's usually for your motorcycle or your vehicle or whatever. But in this case, it's to fuel your body. But chemically, it's very similar. You're just breaking down these carbon molecules. You're releasing energy. We're going to talk a lot about that in chapter 5. Anyway, any questions about this slide? All right. The next word for attendance, sure, will be hamburger. And yes, I know that's a cheeseburger, but the word's going to be hamburger. So again, if you're in person and want the extra credit, make sure you send those words before 9 a.m. If you're online, the live stream, make sure you send the word before 9 a.m. And if you're watching the video that I'm going to post later today, send me a picture of a hamburger, not the word. Find a picture of a hamburger and send it to me. All right. Anyway, if there's no questions about this slide, we'll move forward. We haven't even gotten to the good stuff yet. Your book has a lot of introductory material before we get into the meat and potatoes. You get it? The meat and potatoes were already there. That was actually on accident. So anyway, speaking of which, form and function, this is what octane looks like. And remember I was saying that fat that I showed you looked a lot like an octane, like 3 octanes combined together. And in a sense, that's what it was. Because this is one of those 3 things, right? There's 3 things that are stuck together, and you're going to learn about them later in this chapter. But look how similar they are, right? You can see how they have a similar structure, therefore a similar function. And again, you're going to learn a lot more about it later, but for now, I'll just tell you the function is to break those bonds and release energy. And I said this last chapter, I'm going to say it a couple of times again this chapter, and then starting next chapter, this will be really important information. But for now, it's just an introduction. However much energy it takes to build a molecule, generally speaking, is how much energy is there to be released when you break that molecule. So even without knowing anything about chemistry, you might just guess, because this thing is so long, that it probably took a lot of energy to build it, right? You don't know how hard it was to put this carbon next to this carbon, or to put this hydrogen next to that carbon. But you at least know that it's long, right? So obviously the longer it is, it's going to take more energy to build it. And you would be correct. And that's why one of the reasons why those molecules are so high in energy. All right. So the unique property of these carbon molecules, of these carbon skeletons, they depend on what we call functional groups. And I'm probably not going to ask you the definition of a functional group, so I'm going to put a question mark next to this, because I'm just going to give you examples of functional groups, and that's going to be more important than actually telling me the definition. But just in case, I'll go ahead and tell you. Functional groups are groups of atoms that are directly involved in chemical reactions that are attached to the carbon skeleton. Each of them plays a role in the chemical reaction, and many biological molecules have 2 or more. So again, I'm putting a question mark next to that because I'm probably not going to ask you the definition, because there's going to be so many different functional groups that you're going to need to recognize. Is like, oh, yep, that functional group goes on a protein, or yep, that functional group goes on a DNA molecule. So probably not the definition, but if I do ask you the definition, it definitely won't be that part. So any questions about functional groups? The next slide, I'm going to give you some examples. If you're taking notes, you can give your hand a break on the next slide. I don't even think this next slide I'm going to show you is from your book. I'm certainly not going to ask you to memorize all these different functional groups I'm going to show you. All right. So there's some groups, right? So there's a hydroxyl group is a functional group. Methyl groups, carbonyl groups, carboxyl groups, amino groups, phosphate groups. You're going to be familiar with a lot of these, but you're not going to necessarily need to memorize them. I'm not going to show you a picture of a phosphate group. That's a good one too, because we're going to talk a lot about them, but I'm not going to have you identify them by the way they look. But again, you guys aren't chemists, so you probably can't look at this and know what kind of chemistry it does by looking at it, but you can look at the shapes. And again, things fit together like a lock and a key a lot of times in biology. So the fact that they even have a different shape should tell you that they very often do different things. And then, of course, and I really don't expect you to know this by looking, you can see by looking at the very right, some of them are polar, some of them are nonpolar. So remember, water's polar, right? Because the electrons like to hang out near the oxygen as opposed to the hydrogen, so that makes it polar. Anyway, what I haven't told you yet, and I'm going to officially tell you later, but this is just a hint, polar and nonpolar things don't mix. So if a molecule, I wish I had a laser pointer, if a molecule, some carbon molecule had this attached to it, this hydroxyl group attached to it, and some other carbon skeleton had this methyl group attached to it, they might not mix. They might repel each other. Because polar and nonpolar don't mix, right? So it might be just something as simple as that makes two molecules bend away from each other. And that will be important later. For now, it's just a slight introduction, a little hint of what's coming later. Anyway, any questions about this slide? All right. So let's talk about giant molecules from smaller building blocks. So everything I'm going to teach you, and this is the more important bullet point we haven't gotten to yet, the large biological molecules. Everything I'm teaching you there all basically boils down to this in a sense. So I'm going to teach you 4 different types of large biological molecules. But either way you look at it, they're all big, giant molecules that are made from smaller building blocks. And in the exam, once I get here, you need to differentiate between these 4 large biological molecules. And one of the ways you're going to do that is you're going to need to know what the big giant molecule is called, right? So again, protein, nucleic acids, fats. We're going to learn about that later. But you're also going to need to know what they're called, what their smaller building blocks are called, because they're all different. You know, proteins are made up of different things than sugars. And then those things are made up of different things than fats. So again, for the exam, that's what you're going to need to understand is the difference. One of the differences between those biological molecules. And before we get again, I'm going to give you the generic idea, like just in general, what do we call big things that are made of little things? So any questions about this slide? All right. On a molecular scale, many biological molecules are gigantic, earning them the name macromolecules. There's nothing on this slide I'm going to test you on. There's nothing to write down. Again, this is just an introduction, a segue into the information I'm about to give you. Yes, you will need to know the different categories, but for right now, there's no need to write those down because there's also a 4th category that technically doesn't belong here in this slide, but it does belong in this chapter. So I'm going to put it next to there now. But yeah, these things are technically called macromolecules. I don't need you to know the definition of macromolecules. I don't need you to know that these things are macromolecules and the other one that I haven't introduced you to is not. Only thing you need to know is if I use the word macromolecules, you just need to know I'm talking about big old molecules, right? That's it. That's enough for me. Just understand what I'm talking about when I'm using that word. I'm not going to test you on the definition of a macromolecule. So any questions so far? Okay. And again, I probably wouldn't have even included this yet, but I like to teach out of the textbook, and this is the way your textbook presents it. All right. Macromolecules are something called polymers. You definitely need to know what a polymer is. 1, because I might test you on it, but 2, because I'm definitely going to use the word a lot. And again, like I said, when I teach you those 4 categories of molecules, you need to be able to tell the difference between them. And one of the things you need to know is what the polymers are. So don't write this down yet, but for example, the polymer for DNA or RNA, those would be nucleotides. Polymer for sugars would be polysaccharides. All these things I'm going to teach you later, but you're going to need to know the name of the polymer. So I'm like, here's a sugar. What's the polymer called? Here's a fat. What's the polymer called? Here's a nucleic acid. What's the polymer called? But yeah, so polymers are basically long strings of molecules that are made up of monomers. So you need to know that. You need to know the polymers are the long thing, and monomers are the things that make them up. So you could think of a train as a polymer, and each individual car, or if you call them that, on the train would be the monomer. Or if you have a string of pearls, right? The string of pearls would be the polymer. Each individual pearl would be the monomer. Here's another thing you definitely need to know. What brings these things together? Dehydration reactions. So as far as we're concerned for this entire semester, because this will come up a lot in this chapter and then again in a few other chapters, when I talk about bringing things together and like putting smaller molecules together to make big molecules, those are called dehydration reactions. And I'm not going to ask you this, so you don't necessarily need to know that it removes a molecule of water, but that's why it's called a dehydration reaction, because it removes a molecule of water from the reactants. So remember, chemical reactions are reactants. I'll just put it here. Just to remind you from last chapter, you could have one reactant plus another reactant, and then the arrow, and then let's say P for product. So you need to know the reactants on the left, products on the right in this class. The arrow is always pointing away from the reactants towards the products. So yes, again, it's called a dehydration reaction because it removes a water molecule. Any questions so far? The next slide, I'm going to show you some more information about dehydration reactions. So there we go. There's a short polymer. It probably has a name, but we're just calling it short polymer. It's being attached to a monomer. And of course, it didn't have to be. This could be, it's showing a short polymer being attached to a monomer, but it could be a short polymer attached to another short polymer. Or it could be a monomer attached to a monomer, right? It doesn't matter. The point here is we're bringing two other things together to make something bigger than we started with. And in the process, again, we are basically squeezing a molecule of water from the reactants. Notice that product doesn't have the water. And again, I'm not going to test you on that. That's just how you can remember that that's a dehydration reaction. Another way of saying that is this. I'll give you another generic chemical reaction. If you have a reactant plus a reactant, as many, I don't know, 3 reactants, whatever. The point is, when you have reactants on the left, then the arrow to the right, and the product, if you also have water on the right-hand side with the products, that is a dehydration reaction. And again, not because I'm going to test you on that. I'm just trying to give you hints because I am going to test you on. I am going to give you a reaction and say, was this a dehydration reaction or the other one that I'm going to teach you later? So I'm just giving you hints of ways to identify a dehydration reaction. So again, the ways you can do it is if we're bringing 2 other things together to make something bigger, that is a dehydration reaction. If there's water on the product side, it's a dehydration reaction because we took water away from the reactants. Anyway, any questions about that example? Fair enough. We'll move forward. What's the opposite of that? We don't always build molecules. Sometimes we break them down, especially when we digest things, right? That's the whole purpose of digesting. So we eat these big molecules, and usually we don't need those big molecules. Usually we need the monomers that build them up. Sort of like the Lego example I used a few days ago, where I was like, all right, I give you this big block of yellow and black Legos, and I need you to build me, I don't know, a black Lego car. First thing she needed to do was break down those Legos because they were all yellow and black mixed together. But I asked her to build a black one, so she had to break down and get the parts she needs. Same here. When you eat food, you know, we don't always necessarily need those big molecules, but we do need the building blocks that build them. We break it down, and then we use those parts to build new things. So what do we call when we break it down? The answer is hydrolysis. So again, you definitely need to know the difference between a dehydration reaction and a hydrolysis reaction. And for this entire semester, as far as we're concerned, whenever you're building something, putting something together, that's a dehydration reaction. Whenever you're breaking something down, it's a hydrolysis reaction. And that's where it gets tricky. Had I just left it at dehydration, I think you guys, anybody could memorize that. But then you throw in hydrolysis, and it sounds sort of similar because dehydration, that has to do with water. Hydrolysis has to do with water. So it can get confusing. So I guess the way to help you remember this here is this time, instead of water being taken away from the reactants, this time it's kind of put into it, right? It's almost like if that were a train and we're trying to break it up and they're stuck, maybe you get like some sort of water cannon and you're shooting between 2 of the cars and hoping that that pressure will break them up, if that helps you remember it. So again, there will be some questions on the exam when you have to differentiate between a dehydration reaction and a hydrolysis reaction. So I'll just draw you another one. If we have reactants on one side plus water, if the water is on the reactant side of the equation, it is hydrolysis, right? So that's the opposite of dehydration. Reactants with the water, and then we have the products. In this case, I'll just say 2 products because that's how it's drawn. We have 1 reactant and we have 2 products, A and B. So any questions about that side? All right. Now we'll finally get into the big important stuff. We've given you enough background information to discuss large biological molecules. You need to understand how they work, where you can find them, what they do, what the monomers are called to build them up, what those polymers are called you make from the monomers. You need to know all that for carbs, lipids, proteins, and nucleic acids. And you could write those four down if you want now, but I would say there's no need to because we're going to talk about each of them individually. So again, as I'm teaching this, that's what you need to keep in mind. For the exam, mostly you're going to need to be able to recognize one versus the other. So if I give you an example of a nucleic acid, you should be, oh, yep, that's a nucleic acid. It's not a protein. It's not a lipid. It's not a carbohydrate. And some of the ways I'm going to do it, again, is I'm going to say the monomers of this thing are called this. The polymers are called this. So it'll be a multiple choice exam. What kind of molecule is it, right? So you have to choose of those four based on the information I give you. What's the monomer? What's the polymer? Where do you find it? Stuff like that. So again, there we go again. There they are, the four categories of large biological molecules: carbs, lipids, proteins, nucleic acids. These pictures are not from your book. I just chose them. And these are good independent work topics if you're interested. This is the tardigrade. That's believed to be the smallest animal in the world, and it is tiny. You can look them up if you want to for independent work. Again, they're really, I think they're interesting. You can put them in a vacuum, you can boil them, you can radiate them, and they usually survive. They're really tough little things. And this is, well, actually, what do you guys think this is? What is this a picture of? A forest, yeah, it looks like a bunch of trees, right? And technically that's one organism. So in a sense, some people say that's the largest tree in the world. So anyway, the point that I wanted to get at is like between the largest tree in the world or the smallest animal in the world, no matter what you're looking at, if it's alive, it has these four categories of molecules. Actually, I'm going to share a story with you that I recently found. So we don't have time to get into it in this class, but basically we don't know how these molecules, all the monomers that I'm going to teach you about that make up carbs and lipids and proteins and especially nucleic acids, we don't know how they were formed to begin with. We know how our bodies make up, they put together those monomers to make up DNA, for example, but we don't know how in nature those monomers came to be. But there's been a recent meteor that was found that actually had nucleic acids on it. So there's one theory called panspermia that suggests the building blocks for life came to Earth from outer space, like meteors, and then from there they built up to where we are today. Anyway, let's move a bunch in right there. The next word, because it's 8:36. The next word for attendance will be tree. So again, if you're in person, watching right now, then send me the word tree with the rest of them sometime before 9 a.m. If you're watching the video that I'm going to post later, don't send me the word tree. Find a picture of a tree and send it to me, along with the other pictures. So you won't be sending me any words. Send me these pictures. And yes, I expect your pictures to be different. Anyway, let's move forward. So the first category we're going to talk about are carbohydrates. There's no need to write down monosaccharide, disaccharide, or polysaccharide yet. But that's what we're going to talk about is carbohydrates. What do you see in this picture? What is this a picture of? Spaghetti, yep. What else? Yeah, spaghetti and Coke. And I actually think this is from your book. I wonder if they got sponsored by Coca-Cola. But anyway, what kind of carbohydrates? Where do you think the carbohydrates are in that picture? Let me ask you that. Spaghetti, right? That's usually the number one answer, and that is correct. It is spaghetti. If any of you have ever done long distance running, you're supposed to carbo load the night before, so like maybe eat a bunch of spaghetti. We'll talk about how and why later. But it's not just that, but it's also the sugar in the Coca-Cola. And we'll talk about that as we move forward. But that's a great introduction from your book. So again, carbohydrates include sugars and polymers of sugar. So the sugars would be like the stuff in the Coca-Cola, and the polymers would be the starch in that spaghetti. There's no need to write any of that down. This is still just an introduction to carbohydrates. Here's some stuff to write down. You will need to differentiate between carbohydrates and carbohydrates in animals versus carbohydrates in plants. For the exam, you need to differentiate between the two. For independent work, you could look up the other stuff. Like there's a lot more than just animals and plants in the world. What about bacteria? What about protists? What about fungi? But anyway, in animals, carbohydrates are the primary source of dietary energy, and they are the raw materials for building other molecules. Actually, and let me go ahead and give you a hint too. Like I said, you will need to differentiate between animals and plants when it comes to carbs, but not necessarily this part of it. Once I get to that part, we'll say, all right, here's the big part that differentiates between the two. I'll let you know. But for now, I mean, yeah, again, just an introduction. So that's it in animals, and we'll come back to that. Let's talk about plants really quickly, and we'll also go more into depth about plants. But your book says in plants is the building material for much of the plant body, and that is true. And we'll talk more about that later too. But what your book didn't point out, either that or I somehow accidentally left it off and didn't put it on the slide. But carbohydrates are also an energy source for plants too. As a matter of fact, when we start talking about photosynthesis, you're really going to get familiar with that concept because that's their energy. Yes, they get solar energy, but the same way our cells need carbohydrates for energy, like I just said up here, so do the plants. They don't even use the sun's energy directly. They take that sun energy just to make carbs. They build up the carbs and then just break them down the same way we do. So, but we'll get to that much later. Any questions so far? All right. So again, for carbs, one of the things you need to do is understand the 3 different types of carbs and be able to pick them out of a lineup, so to speak. Understand the differences and similarities between these things. So here we go. And the names kind of make it easy. First one we're going to talk about is monosaccharide. And those are the actual monomers of carbohydrates. So like I said earlier, you need to understand the monomers and the polymers. Right? So that's going to be some of the ways you differentiate between carbs and proteins and lipids and nucleic acids. So I'm telling you right now, the monomer for carbohydrates are called monosaccharides. You need to know that. So I'll circle it. And I'm going to put an X to the next one, not because you don't need to know it, just because I'm not going to test you on that. The point is, if you break down the monosaccharides anymore, like if you do anything to it, if you break it down anymore, it is no longer considered a monosaccharide. That's what that means by it can't be broken down into smaller sugars. So for example, you're going to learn about C6H12O6, right? If you break that down anymore, it's no longer a sugar. It's no longer a monosaccharide. So again, monomers are basically, again, just the parts that make up the polysaccharide. In this case, they're called monosaccharides. And they can't be broken down into smaller groups. Here's some examples. Glucose. So when you drink things like Gatorade, that has glucose in it. When you drink things like juice, like fruit juice, that's usually fructose. If you eat honey, it has both of them in there. Of course, these are just examples. I'm not gonna test you on fruit juice or Gatorade or honey. But anyway, again, for the exam, the most important thing on the slide is you need to know that for carbohydrates, the monomers are called monosaccharides. So here's some examples. Again, if we're talking about honey, you can find both glucose and fructose. And here's some more interesting stuff when it comes to form and function. Again, you guys aren't chemists, but I told you things often fit together differently, right? And a lot of times that's what chemistry is. They have these molecules fit together. And you can see here, these things even have the same formulas. Glucose, C6H12O6. Fructose, C6H12O6. Same chemical formula. Matter of fact, you're going to draw that in lab if you haven't already. And what you'll notice, if you're really paying attention, is that there's different ways to draw the same chemical formula. This is a great example. And that is what an isomer is called. And you need to know what an isomer is. Same formula, different arrangement, different structure, however you want to say it. Of course, glucose and fructose are not the only isomers in the world. That's just one example I'm giving you where you have the same formula, in this case, C6H12O6, a different structure. But anyway, like I was getting at, you can tell by looking that these things have a very similar structure, but slightly different. And that is enough to tell you that they have a very, very similar function, just slightly different. You might not be able to tell the difference between glucose and fructose when you taste it, but you probably could. It's subtle, but fructose is sweeter. All that because it's a slightly different shape. Anyway, any questions about this example or what an isomer is? And again, I'll point at this. As far as the exam is concerned, that's the important information. You need to know what an isomer is. I think the next slide is another example of an isomer that's not in your book. No, not yet. We'll get there. But yeah, so what I just taught you, this is just the slide where it's officially on the board. Glucose and fructose are isomers. Isomers are molecules that have the same molecular formula, but a different structure. So yes, you need to know that. And obviously not verbatim, because as you can see, I kind of wrote it two slightly different ways. But so when you're studying definitions, don't just study exactly what it says. Study it in your own words if you want. All right, moving forward. Yeah, here's a non-book example. I don't expect anybody to know this, but you can see, right? They look very similar. They're like mirror images of each other. They have the same formula, but they're slightly different structure. Does anybody know what these are? I mean, you can kind of get a hint if you read it, but anybody know what those molecules are by any chance? I don't expect you to. Just an example. The one on the left is basically a menthol, right? It's the stuff that you would find in Vicks VapoRub or even in menthol cigarettes, right? It's that stuff that kind of clears you up. And the other one, just by making, like, again, same structure, just a mirror image, it's meth. So that goes to tell you the difference between, you know, how isomers can be such a big difference. And that's also a good independent work topic if you want to look it up, if you want to look into it. Some of the interesting isomers out there, because some isomers, like, some of them are good, some of them are deadly, right? So they have the same chemical formula, but if you change the slight way that those things are put together, sometimes they can kill you. Anyway, obviously, I'm not going to test you on meth or Vicks VapoRub. That's just a non-book example. All right, monosaccharides, especially glucose, are the main fuels for cellular work. I'm going to put an X through that only because, actually, no, I'm going to put a big L through that. Not because you don't need to know that, but you don't need to know it for this exam. I mean, it's a good introduction, but you're going to be really familiar with monosaccharides when we talk about chapters 5, chapter 6, chapter 7. You're going to know all about monosaccharides, especially one particular type. Also, and this isn't important for the exam, but this will be important later, so I'll put another L next to it. In water, a lot of times those monosaccharides forms rings. So let me back up really quickly and just show you again. Glucose and fructose, I just showed you, look like that, right? They look like long strings, and they are. But a lot of times when you put them in water, they form rings. And the only reason that's important for this conversation is because, remember, I said they are monomers, right? So a bunch of them, you put a bunch of them together to make polymers. So before they get linked up, it's not like they're just a bunch of long rings attached to each other, or long strings. They turn into rings, and then the rings, and they attach the rings together. You'll see what I mean when I show you a picture. That's what I mean, right? So when we're talking about, right, again, that's a monomer. So if we were to make a bunch of monomers, put a bunch of monomers together, I'm not going to string them up like this. Like attach one here, attach one up there. That's not how it works. Not that I'm going to go this much detail in the exam. But before they get linked together, they form rings like this. So what we have, as you're going to see later, is when we put these monomers together, it's this ring is going to be attached to another ring, which should be attached to another ring. So that's how it works. And for the sake, for the exam, that's not that important. Also, even if you forget that, you can still understand the information I'm going to give you later. So this is relatively unimportant information at this level of biology. Whew. So anyway, any questions before we move over to disaccharides? Any questions about what monosaccharides are? So again, those are the monomers that make up the polymers, right? We put those things together to make larger carbohydrate molecules. So before I get into disaccharides, does anybody want to take a guess? How many monomers make up a disaccharide? Good, right? I'm glad you guys are familiar with your prefixes. Yes. Disaccharides are double sugars. They're constructed from two monosaccharides. As far as I'm concerned, that's pretty much the only thing you need to know for the exam. There will be some more questions on the study guide. Here's some examples. I really don't think I'm going to ask you about any of these examples. If I do, it might be this one, and I'll come back to that here in a couple slides. But yeah, sucrose, lactose, maltose, just some examples. I'll talk about them. I'm probably not going to ask about them. Again, you can look at them and say, oh, yeah, those things look very similar, but slightly different. Likewise, they have very similar functions. They're just slightly different. And I'm going to come back to, again, some examples. So for right now, are there any questions about that first bullet point? What a disaccharide is. All right, if you download the PowerPoint, you can watch two monomers coming together, two monosaccharides coming together to form a disaccharide. Here's what it looks like in drawing. If you put glucose and galactose together in a dehydration reaction, right? It's always a dehydration reaction when you bring things together, building something bigger. That gives you lactose. Anyway, that's just an example. I'm not going to ask you about lactose. I'm not going to ask you what it's made of. Again, the only thing on the exam that you should recognize when you see this picture is like, oh yeah, that's a dehydration reaction because it took water out of the product, out of the reactants. And we started with two monomers and ended up with a polymer. Some examples. Not that I'm going to ask you any examples, so I'm going to put an X to this to indicate that. But some examples include lactose in milk, maltose in beer. So when I'm going to study for this tonight. Maybe drink some beer. I'm just kidding. But anyway, maltose is in beer. Malted, so you've got things like malted milkshakes, or what are those candy called? They're covered in chocolate. Whoppers, maybe. Malted milk ball candies. I think they're called Whoppers. Anyway, those are just some examples. Sucrose and table sugar. So when people say sugar in a non-scientific term, that's usually what they mean is sucrose. And sugar will be the last word for attendance. So when you, again, if you're watching the video, send me a picture of sugar. And that's it. So I guess if anybody wants to stick around and do a picture, go ahead and do it. If anybody wants to stick around and volunteer to take the picture, we'll do that. But that is it. I will see the rest of you who weren't there on Monday. I will see you in lab at noon. So Reed and Felder, did you need anything? Yellow. All right. Well, if you need me, I'll be here.